Okay, without further delay, I shall continue with the sharing session on today. Okay. So the agenda for today's sharing will be including number one, what is gene synthesis? And secondly, the application on the synthesis part. Next, we shall also touch on how does gene synthesis being performed, and then we'll look at if you are interested to to go for a gene synthesis service, what are the requirements or how you actually want to get a quotation from us. Next, we'll look at additional offers from GenScript related to gene synthesis. And we shall end today's session with some frequently asked questions about gene synthesis. Then after that, we shall do some discussion if you have any question. Okay, so let's move on to the first agenda of today, which is what is gene synthesis. So by definition, gene synthesis refers to chemically synthesizing a strand of DNA base by base. Unlike DNA replication that happens inside a living cell or amplification inside a tube by using polymerase chain reaction, gene synthesis is a method or technology that does not require a template strand. So basically, it's considered de novo. Gene synthesis involves the stepwise addition of nucleotides to a single-stranded molecule, which then this particular single-stranded molecule will serve as a template for the creation of a complementary strand. Gene synthesis is actually a fundamental technology in the field of synthetic biology. Okay, so next, our question will be, what can be synthesized? Artificial gene synthesis is used to chemically synthesize a DNA sequence, and this DNA sequence normally representing a gene. Let's say you have a gene of interest from one particular organism, or even multiple genes. The gene synthesis technique provides a method that efficiently produces long stretch of nucleic acid sequences. Okay, this particular method is actually particularly useful when you have sequences that are hard to come by. For example, let's say you have a DNA sequence that is hard to isolate from the natural sources. By using gene synthesis method, you can produce this particular sequence routinely in your lab. Or sometimes, if you have a variance of a gene that you want to do comparison compared to the wild type, and this variance might be tedious to produce through site-directed mutagenesis, then you can sought the help of gene synthesis. And actually, there are no restriction on this particular sequence. You can actually you can synthesize sequence that don't even exist in nature. Here we shall look at some of the benefits of gene synthesis when we are in different fields. For example, gene synthesis has been used in the field like research field, healthcare, enzymology, agriculture, environmental, and metabolomics, to name a few. In research, synthetic genes are used in diverse biological studies, okay, including but not limited to encoding of proteins and then using the protein to regulate gene expression. Or you can actually use gene synthesis to express protein that mediating cell-cell communication. Those proteins can be used to study the formation of biofilm as well. In healthcare, in some cases like the recent event, the COVID pandemic, we can quickly synthesize newly identified viral genome, and this will help to accelerate the vaccine development. For enzymology, we can engineer novel enzymes that fight cancer, or for commercial use. For example, produce sustainable biofuel. It can also help and useful in agriculture because we can improve crop yield and reduce vulnerability to the common pests and plant diseases through transgene. In environmental field, we can produce protein that detect and break down environmental pollutants. And in metabolomics field, we can actually design and build the metabolomic circuits using the synthesized parts. Traditionally, we can use PCR and molecular cloning method to achieve to get the gene of interest. However, there are some limitations to this method. Number one, it's limited to sequences that appear in nature. And secondly, most of the time, codon optimization is not feasible. We shall talk about codon optimization in the later slide. It is also time and resources intensive for end user. In comparison, gene synthesis with subcloning to a plasmid vector offers customizable sequences. Okay, and this means that no template actually needed to be provided by the user. It can uniformly accurate sequences from a clone. And it's easy to verify complete sequences using primers that flank gene insert. It is a fast and cost-effective method. So the main point here is the de novo chemical synthesis of a DNA, which is gene synthesis. It does not require a template, and it allows researchers to specify a desired sequence and custom build it directly. Gene synthesis is frequently far more straightforward and faster and less costly compared to alternative methods. Here, I would like to share on the type of research application of the gene synthesized part. Some of this application includes you can synthesize a PCR product and then clone it inside a plasmid vector. Then this particular control, the plasmid with the insert, will be used as a PCR positive control. In addition, you can actually synthesize several parts of a viral genome and then use it as a viral genome positive control for next generation sequencing. Another application will be like to create a template for in vitro transcription. Again, it's something to do with the virus pandemic. You can use it to produce, let's say, mRNA vaccine. So for mRNA vaccine, you actually create a DNA plasmid template, and then you use a method that called in vitro transcription to produce the RNA. For more commonly used application, we will synthesize the ORF, which stands for Open Reading Frame of a gene. For these coding sequences, we will clone it into a plasmid, and then we will perform overexpression. It can also be used, the synthesized gene can be used for reporter assays. We shall talk a little bit more on this in the later slide. And it can also use to create CRISPR required parts or components. And it can also use for in the experiment of knockout experiment. Okay, so here I would like to zoom and go into further details for some of the research application that I mentioned earlier. So here we will share on using gene synthesis for recombinant protein expression. In this particular application, we will be using a plasmid, and the plasmid is not integrating into the genome. Means that it's a free plasmid that do not join or become part of the genomic DNA. Sometimes we refer to this kind of expression as episomal expression. Inside the plasmid, we will have the gene synthesized coding sequence that we refer to CDS. Also corresponding to the open reading frame of a genes, basically is amino acid coding sequences. So inside this plasmid with the CDS, we can actually go for overexpression or heterologous expression. Overexpression means that we produce the gene, the protein of this gene of interest in large quantity. And for heterologous expression, it actually means that we express the protein in a non-native host. Let's say the protein originally is from human, and we express the protein, the gene, in E. coli, then it's considered heterologous expression. Synthesized CDS can encode either naturally occurring transcript or custom design variants such as point mutants or fluorescently tagged reporters. Gene synthesis accelerates the creation of recombinant coding sequences such as fusion proteins or promoter-reporter constructs compared to traditional molecular subcloning and mutagenesis techniques. Protein expression level can be greatly improved through codon optimization, which is simple to perform as part of the synthetic gene design, but practically impossible to perform using traditional recombinant DNA technology. Synthetic DNA can be cloned into expression vectors and used in any protocol that require natural or recombinant DNA. So just now we are talking about the standalone plasmid. Here we might need the plasmid or gene to integrate into the genomic DNA. The difference is that in mammalian cell, if the gene is integrated into genomic DNA during multiplication or mitosis, the daughter cells will actually contains the modification as well. If you are using episomal plasmid, the plasmid will be lost after several multiplication. So here we shall go into genome integration, where the genomic DNA will have the synthesized gene parts. So synthetic genomic DNA can be studied in vitro, inserted into whole cell in culture, or used to create transgenic animal lines. So either through gene knock-in, means the insertion of a gene, or gene knockout, or the deletion of a gene. Conventional gene targeting of embryonic stem cell with disruption cassette inserted through restriction enzyme or Krylog-P system. Simpler, more rapid, and less expensive by using synthetic DNA in combination with the newly developed methods for gene editing, such as CRISPR-Cas9 nuclease, zinc finger nucleases, or transcription activator like effector nucleases. So if you are doing related study or you are interested to perform like gene editing, gene synthesis techniques or method could be useful to speed up the whole process. Okay, here we shall take a look at what exactly is codon optimization and whether we should perform codon optimization or not. Okay, so before we are making a decision whether we want to perform codon optimization, let's take a look what it is and why we want to do it. So in order to characterize a gene's function or purify a protein for structural study, researchers frequently need to express a gene in a host cell, means that non-native host, or model organism different from the one in which it appears in nature. Basically, in short, heterologous expression. Okay, unfortunately, heterologous expressed gene often suffer from low rates of protein expression. Means that we want to overexpress the protein, but the problem is we are getting a small amount of this protein of interest. A common cause of low expression levels is the variation in codon usage frequency between different species. This can cause the translation of heterologous expressed gene to store due to tRNA scarcity, leading to lower protein levels and increased rates of improper protein folding. The degeneracy of the genetic code make it possible to change the DNA sequence in a way that does not alter the final amino acid sequence, but does have significant effects on the efficiency of transcriptions, mRNA stability, translation, and protein folding. Aside from codon usage bias, many other features of a DNA sequence affect the efficiency of transcription, the proper splicing and processing of mRNA, and mRNA stability, all of which will in turn reduce the ultimate protein yield. So therefore, a codon optimization is actually modifying a DNA sequence to optimize it for efficient protein expression, at the same time preserving the amino acid sequence. So in a study by Burgess Brown, actually a 2008 study, they investigate the expression of 30 human genes in E. coli. They compare the result of codon optimized sequence versus the native sequence, and they found that the total protein yield and solubility is a lot better increase for the optimized sequences. Okay. And since then, the algorithm has been improved and now widely used by researchers to facilitate the purification of protein for various studies. Okay, so in GenScript, we are offering a free codon optimization service for all gene synthesis project. Okay, and it's actually named as Optimum Gene. Okay, other freely available codon optimization tools include JCAT and Optimizer. So next, we'll move on to another application of gene synthesis in research, which is actually mutagenesis. Okay, so gene synthesis can facilitate the discovery of new variant, okay, to be used in different fields. Okay. Traditional method for inducing random mutation will use like living cell or animal and then expose them to UV radiation or chemical mutagen. So the effect is UV radiation and chemical mutation will induce DNA damage. Okay. Inside the living cell, DNA repair enzyme, in some cases, will try to repair the DNA damage, which introduce or preserve mutation from the exposure. However, the UV method or the chemical mutagen method leads to high rates of fatality. Okay, and actually has low rates of successful to produce useful mutant phenotype. Okay. Therefore, in research, most of the time we are looking for like site-directed mutagenesis. Okay, so we are actually focusing on a point to create a single nucleotide polymorphism, or small deletion, or small insertion. So the design mutation or alteration is based on experimental data such as X-ray crystallography data that identify the base of interest. Site-directed mutagenesis can be performed using PCR-based method with mutated primer sequences to create and selectively amplify mutated sequences. This type of mutagenesis must be followed by sequence verification and could be tedious, error-prone, and costly. Okay. The next research application or how we want to use the gene synthesized parts is for reporter assay. Reporter assay in general, we are using it for two applications. The first one on the left here is actually used for promoter study. Basically, we gene synthesize a promoter sequence and then we fuse it in front of a luciferase gene. When we transform or transfect this particular plasmid into our whole cell, we will actually treat the cell with a proper inducer or agent to see whether it activated the promoter or not. If the promoter is activated, luciferase mRNA will be produced and followed by luciferase enzyme. With the addition of the substrate, we will detect a luminescent light. Okay, this shows that the agent or treatment that we added, induced, is actually activating the activity of the promoter. So in another experiment, we are using similar setup, but in this particular study, we are actually looking on the interaction between microRNA and the 3' untranslate region of mRNA. We try to see whether the microRNA is binding to our mRNA of interest, and if this happens, most likely is going to degrade this particular mRNA. So therefore, we are studying the interaction between microRNA and the 3' end part of messenger RNA. Okay, the promoter is not modified in this particular experiment, different from the earlier one. Okay, so we are modifying the 3' untranslate region. We actually gene synthesize and clone it over here, the green color, and then we actually transform or transfect these two things into the cell. And if the microRNA is interacting with the 3' UTR region of this construct, okay, it will actually degrade the whole microRNA, and as a result, you will not detect any light or luminescence. However, if the microRNA does not interact with the 3' UTR, you will get the signal. So next, I will move on to the next topic, which is how gene synthesis perform. Okay. So basically, the basic steps of gene synthesis are sequence optimization, okay, DNA sequence selection, and oligo design. Basically, we are synthesizing some short oligo. Okay, so therefore, we will actually go to oligo synthesis. Then we try to assemble and join all these short oligo together into a longer fragment, which is the gene of interest. After the assembly, we will perform sequence verification and error correction. This can be achieved by using technology like sequencing, okay. And next, we will be able to produce the synthetic DNA and deliver it to the user for downstream application. Okay, so there are many methods for gene assembly. Okay, so some of it involve the use of polymerase, and another technique or method will be using ligase-based in vitro assembly method. Okay, so the correct assembly required high fidelity enzyme, and polymerase chain assembly method (PCA) is a standard technique for polymerase-based oligo assembly in a thermocycler. Okay, so this reaction is actually templateless, and the principle is to combine all the short oligos originated from different part of the sequence of interest into a single tube. Then we perform some temperature changes to facilitate repeated rounds of annealing, extension, denaturation. And then we use the outermost primer to amplify the full-length sequences. After that, we shall go for sequence verification and error correction. Okay, because the step might create incorrect amplification, we will need to perform some correction and sequence verification. One of the things that we can do is we actually clone it into a plasmid vector, and then we sequence the plasmid. Okay, because each plasmid should only harbor one sequence, and we will actually screen the clone for the correct gene construct or gene sequence. Okay, so this is showing a result of gene synthesis sequence verification using Sanger sequencing. We are comparing the sequence of interest versus our sequencing result. Okay, so we will be providing the AB1 file as well, and also the comparison report. Okay, so for most applications, the synthetic gene will be cloned into appropriate vector. Okay, this may include plasmid vector for transfection or electroporation into cell. Okay, so facilitate cloning, synthetic gene may be designed to include restriction enzyme sites, recombination arms, and other flanking sequences. As an alternative to avoid introducing special flanking sequences, recombination-based method begins with PCR extension of the gene insert to introduce 15 base of sequence homology to the linear vectors, facilitating homologous recombination without adding unwanted base. Okay, so this is another showing another QC step, the quality control step in the certificate of analysis report from GenScript. In addition to Sanger sequencing, we will also use restriction enzyme to digest the plasmid and see whether it releases to fragments that with expected size or not. So after we get the genes construct, we will actually propagate it in E. coli. And the propagation sometimes may be challenging for several reasons. One of it will be the low copy plasmid construct does not amplify well in the bacterial host. And a lot of times this is caused by long genes, because long genes are often difficult to maintain and propagate inside the host E. coli cell, because it causing energetic burden. Some genes may alter the physiology of their host, creating aberrant culture temperature requirement or other conditions that may be difficult to identify. So accommodate genes the synthesized gene may also be toxic or unstable in certain host cells. However, it does not, in some cases, in some host cells, they can tolerate this. Okay, so therefore, screening a number of cell lines to find the best host can be done, okay, but it might be time consuming. And in GeneScript, we are offering some whole cell pool that can determine the best bacterial host for propagation for difficult genes and deliver a vial of this cell with purified plasmid DNA. Okay. So a little bit more about copy number. So a lot of times is actually decided by the origin of replicate in the plasmid. So for example, these are some of the name of the origin of replicate of plasmid. Okay, so let's say this is the plasmid name PSC101. Then this is the origin of the plasmid, and we actually decide the copy numbers. Basically, low copy number is the number of plasmid inside a host cell. Okay, so low copy number maybe only have like 15 to 20 copy of the plasmid inside a host cell. As for high copy number plasmid with the high copy number origin of replicate, we have around few hundreds of copy of the plasmid inside the host cell. So next, I shall continue with if you are interested to go for the gene synthesis service, what is required to give to us next gene to generate a quotation for you. Okay. Some consideration factors for looking for gene synthesis service. First thing is we will need the user to provide the sequence inside a text file, inside a Word file, or in some other FASTA related, or even like accession number from the NCBI. Okay. Next, we need to decide because gene synthesis service, we will not provide double-stranded DNA to the user. We will actually provide the double-stranded DNA clone inside a plasmid, and then we will deliver that to the user. So therefore, the choice is that in standard sense, we will provide and use the vector PUC57. Okay. So here, this one is actually showing the gene sequence. Okay. So you can just give us a text file containing the gene of interest. Okay. Also, it's possible for you to give us just the protein sequence. Okay, like the one down here. Okay, so the standard vector PUC57 is shown over here. This is the vector map of the PUC57. The size is 2579, and we will actually clone your gene of interest or synthesized gene inside this particular multiple cloning site. Okay, if we zoom in the multiple cloning site, we will see these sequences on your right. And over here, if the user did not have any specific requirement, we will actually open up the equal R5 side of this MCS, and you generate a blunt end. Okay, and the gene of interest will be in between. Okay, it will be the continuation of this sequence. Okay, over here, you continue with the gene sequence, then continue with the plasmid sequence, and then form a round shape. So next consideration will be, do you want to include a restriction enzyme recognition site for subcloning purpose? Basically, after we deliver this particular vector with your gene or sequence inside here, okay, do you plan to transfer the gene fragment out, or you want to release it from this vector or not? Okay, so for the application like PCR control, most of the time you do not need to release it or to remove the backbone or transfer it to another expression vector. In that sense, it's okay not to include any restriction enzyme site in front or after your sequence of interest. Okay, for example, this is your sequence of interest. Okay, this is the addition of a restriction enzyme site. So the purpose or function of this restriction enzyme site is so that after you receive the PUC57 with your sequence of interest here, you can use the enzyme to digest and release this fragment from the vector backbone. After that, you can actually subclone it to another vector or for some other application. Okay, so this is the RE site selection for subcloning. Okay. Let's say the user is planning for subcloning because they want to transfer the sequence of interest into an expression vector. Okay. For this, in GenScript, we are offering something called express cloning. So not only we offer the synthesis of the sequence, okay, that supposedly is delivered in the PUC57, we can help the user to transfer this gene sequence to a vector of choice. Okay. So these are the expression vector in GeneScript that we are offering a lot of time FOC, but in some cases it's actually charged, but most of the time is FOC. Okay, so you can select which vector you want to subclone your gene sequence into, and then next you can straight away perform your expression. Basically, you transform or transfect this vector with your sequence of interest into your whole cell, and then to express the protein. Okay. So therefore, in some cases, if the question is, how do I choose the cloning site? Basically, it actually depends on the expression vector. First, we need to select the expression vector, and then we look into the multiple cloning site. So this is the zoom view. Then we will decide which restriction enzyme we want to use, and then to excise it, to digest it, and then after that, continue with our gene sequence. Okay. So next, maybe you are asking like, how do I choose the expression vector? Okay. So which expression vector should I use? There are so many. Okay. The first thing we need to decide will be like, are we going to use a mammalian host, a yeast host cell, insect cell line, or a bacteria? Okay. So these are the some consideration, and we shall continue with this topic in the FAQ session. Which host cells to choose? Okay, so here I was just showing you if you are looking require a codon optimization in GenScript. After we using the optimum gene analysis, we will generate a report for you, and inside the report we will compare the sequence before and after. Okay, and then we will try to avoid your restriction enzyme site within the gene sequences. Okay. So next, I shall share with the additional of gene synthesis, okay, and also subcloning. Okay. So we offer glycerol stock. Okay. In standard delivery, after the gene synthesis service, we deliver the gene within a vector. Okay. And this is actually a plasmid DNA, dry form. Okay. In some cases, if you need to get more, because we deliver the final construct normally in the sense of 4 microgram. Okay, if required, if you need more, you can actually request for glycerol stock. Okay, there will be a small charge. Okay, then in addition to the standard delivery of the lyophilized plasmid DNA, we will deliver the E. coli containing your plasmid of interest in this strand. Okay, so we deliver this E. coli strain with your plasmid of interest. Then you can propagate and do plasmid extraction to get more of your plasmid DNA. Okay. If you do not wish to perform the bacterial propagation and plasmid extraction, we do offer plasmid extraction service as well if you need more of this plasmid of interest. Okay. In addition to that, we also offer low endotoxin grade plasmid DNA in case you are doing mammalian cell culture that are sensitive to endotoxin. Okay. Another added features from GenScript will be for all your vector or your final product, we offer vector storage. Okay, so you can actually store it for up to 5 years. Okay, and if for your next project you would like to use or modify your previous project construct or plasmid, you do not need to deliver us anything, or you do not start from scratch. We can continue from your previous project. Okay, so now we will go to the FAQ session. Okay, so the first FAQ favorite asked question will be, is my synthesized gene accurate? So we actually guarantee the sequence accuracy, and we will use the Sanger sequencing method to confirm the sequence accuracy. Okay. The important point is that when we generate the quotation to the user, okay, we will deliver a text file to the user. So the user need to confirm that the sequence inside the text file is accurate. So the end result or the Sanger sequencing result will be comparing to this particular FASTA file or text file that come along with the quotation. Okay, so we'll guarantee the sequence accuracy compared to the agreed sequence. Next, the common asked question will be, how much is the gene synthesis service? Okay, so actually the service is length dependent. The longer it gets, the more expensive it will be. Okay, however, we will require the actual sequence to provide the actual price. Okay, No worry, when you're requesting for quotation, we do not charge. So therefore, you just send us the sequence of interest. We shall generate you a quotation for free. Okay. And next, what is the turnaround time? In general, with the quotation, we will actually mention as well the duration that we need to deliver the sequence. Okay. In general, we need around 28 to 33 business or working day, not including public holiday or Saturday and Sunday. Okay. This one normally is the time required for gene synthesis as well as plus gene synthesis plus the subcloning into the vector. Okay. If you need the final product urgently, GenScript also offer fast service, of course, with additional charges. Okay, here we shall continue to our discussion earlier. Okay, which expression vector to use? Okay, so just now we see that in the vector, free vector or IP vector offered by GeneScript over here, the main title we have mammalian cell is insect and bacteria. Okay, so therefore the first thing we need to decide is which cell we want to use. Okay, and this graph, this table basically is comparing between different whole cell, the advantage and disadvantages of using this different whole cell. So for E. coli, the good thing about E. coli is the cost, the cultivation to grow the E. coli is very low. And you can easily get a lot of E. coli containing your plasmid and protein very fast because the doubling time, the multiplication of the E. coli is very fast, 20 minutes. Yeast cell is slower to increase in number. And of course, mammalian cell is very slow. Okay, so therefore the productivity of recombinant protein for E. coli whole cell is very high. This one is low, high. This one is okay. However, there are some advantage of using yeast cell and mammalian cell. For example, they are able to create disulfide or post-translational modification to your protein of interest. Okay, or for mammalian cell, we can even have post-translational modification like glycosylation. Okay, this all will help in the protein folding into the native form. Okay, let's say if you are using a protein from eukaryotic cell, okay, if you want proper folding, the best bet will be using the mammalian cell. Okay, however, you can always use E. coli to do a test run. Okay, you can also find some literature to see whether that particular protein will be able to fold properly inside the E. coli cell or not. Okay. Otherwise, some test and error might be required. Okay. After we are selecting the whole cell, then we can go through some common expression vector that we can use. Okay. Basically, here for mammalian cell, the most common expression vector will be the pcDNA 3.1. It's a very wide, commonly used, and a lot of researchers are using this for the expression inside a mammalian cell. Okay. So for bacteria, the most common one will be the PET. Okay. So there are so many PET, but you can ask us for some recommendation, and we shall tell you which one we recommend and what is inside the expression vector, why we actually recommend this particular vector. Okay. So the different plasmid, basically they are using different promoter. Okay, so we have the T7 promoter in the PET or bacterial expression plasmid. Okay, so this one is to work together with the host cell, the bacteria E. coli. Then for mammalian cell, most of the time we are, let's say in the pcDNA 3.1, we are using the cytomegalovirus CMV promoter. Okay, so this will have a high expression by using this one. And if you are using plant cell, although GeneScript did not offer any plant plasmid or vector, okay, they are actually using another promoter called, let's say, 35S cauliflower I forgot what's the M stand for, but cauliflower something, mosaic, I think, yeah, cauliflower mosaic virus promoter. Okay. So next question you might be asking is, can I use my own vector? Let's say you construct your own vector, or you get your vector from your collaborator, or the vector of that you prefer to use does not offer by GenScript. So can I use my vector? So the answer is yes, you can use your own vector, but because we need to deliver your vector to GeneScript, so we will actually incur the transport charge to deliver the vector to GeneScript. Okay, so what restriction enzyme site should I use? Okay. So over here, a lot of time we decide the restriction site in the multiple cloning site. So first we need to choose the host. Secondly, we need to choose the correct vector. Let's say in this particular sense, we are trying to express protein in E. coli. Therefore, let's say we are using PET32A. Promoter, which is the T promoter. A lot of times we need the element ribosome binding site as well. Therefore, our protein coding sequence is after RBS. Okay, you can see from the illustration here, we only see the protein coding sequence, methionine, the first amino acid, is right after RBS. So therefore, when we're choosing our cloning site, it should be, for bacteria, it should be after RBS. Okay, next, when we are selecting the cloning site, our consideration, do I want to retain this particular TRX tag or not? This tag is already inside the plasmid vector. Okay? So if I need his tag, then, and also the S tag, we shall look for RE after that. Let's say, for example, BGL2 or KPN1. If I do not want his tag or S tag, then I should choose, let's say, MSC1. So after choosing the first IE site, okay, to decide the 5' end of our sequence, we also need to choose another IE site for our 3' end. Let's say if you are choosing MSC1, okay, and then we choose, let's say, XHO1 for our 3' end. Okay. So the sequence of the vector from MSC1 until XHO1, this particular middle part, will be gone. Okay. So starting from MSC1, we'll be continue with our coding sequence, and then straight away until XHO1, then continue with the histone. So when we are selecting the multiple cloning site, when we are selecting the restriction enzyme, a lot of times you might get a comment from, let's say, us, NextGene or GenScript. We'll talk about this might create a frame shift. Okay, so what exactly is the frame shift? Okay, so I have here two sequences of that are the same. Okay, so I'm trying to clone a gene using the NCO1 cloning site. Okay, so therefore before the NCO1 is actually the vector sequences. Okay, so these are all the vector sequences, the promoter, the RBS, the 6S, the S tag, just exactly like the PT32. Okay, it's all from the vector. Okay, so I would like to choose an IE site NCO1, then continue with my sequence of interest. Okay. So in this particular example, if let's say the NCO1 is it's actually CCATG. CCATGG. Okay, NCO1 is the highlighted area, CCATGG. Okay. So the problem of choosing NCO1 is that the code is not in continuation with our gene, because our gene start with ATG, then AAA, AAA. We want the ATG to be coding for methionine. However, the NCO1 CCATGG, okay, CCA is not coding for amino acid. Okay, instead, the CC of the NCO1 is the continuation of G. So GCC means two bases of the NCO1 is actually coding with the A, okay, together with the G base. So if we directly put our ATG right after NCO1, the frame will be shifting. Okay, instead of ATG coding for methionine, we have the G base left over from the MCO1 continue with the AT. So GAT will be coding for D. Then after that, all our coding sequences will be all gone, and then it actually create a premature stop codon TAG, just like 1, 2, 3, 4, 5, 6, 7. After 7 amino acid, you actually create a stop. Okay, so this is what we call a frame shift due to the restriction enzyme that we choose. Okay, to try to solve this problem, okay, our suggestion will be to add additional 2 bases in front of our gene of interest. Okay, so in front of our gene of interest, ATG, the start codon, we add, let's say, 2 bases CT. Okay, so the NCO1 will be ended with GG. Okay, so one of the G will be single out and continue with the GCT that we added to create additional amino acid A. But by doing so, we will actually get the correct coding sequences, the frame of our gene of interest. Okay. Okay, next question will be, do I need to do gene synthesis again for my variants? Let's say I have my wild type gene, okay, so let's say it's 1005. Then I need to do a single point mutation to my variance mutant. Do I need to synthesize another 1500 bases again or not? Okay, we know that just now, as mentioned earlier, the cost depends on the length of the sequences. If you want to go over another 1005 gene synthesis, it will be quite costly. Okay, therefore, for this kind of example, we are offering the mutagenesis service. Okay, so instead of charging the full price of the 1500 gene synthesis, we are offering the mutagenesis service where we only performing point mutation. Okay, so let's say SNP that is within 40 bases. Okay, so if you have any modification SNP you would like to change over here. Okay, so this is the wild type. Okay, this is the mutant. Okay, so if you want to change any sequence within this region, okay, you can do it using a single mutagenesis service. Okay, or if you want to delete this sequence, no problem, we will charge one mutagenesis sequence. Or if you want to insert additional 40 nucleotides further over here, no problem as well. We only charge it for a single mutagenesis service. Okay, you do not need to synthesize the whole thing over again. Okay, so with that, I shall end my session today for the gene synthesis service, the seamless cloning gene synthesis service from GeneScript.
Thank you so much, Yifeng, for the wonderful presentation. Now, we will go ahead with the Q&A session. Please feel free to voice out if you have any question by unmuting your mic, or you can type in the chat box. So in the chat box, we have several questions. The first question from HL Wong. Can the user specify the RE sites to be inserted in the expression vectors? What are the charges on the expression vector? Can GenScript provide cloning into a gateway cloning vector?
Okay, so thank you, Wong, for your question. Okay, can the user specify the RE site to be inserted in the expression vector? Okay, so the thing is, we need to have a shared RE site of our gene synthesized sequence and the RE site inside the expression vector. Okay, so we need to share so that when we do like subcloning, we can actually use IE to cut, okay, to release our gene of synthesized gene fragment, okay, and then pair it with the same sequence that we cut in the expression vector. So therefore, let's say if in the expression vector you have, let's say, KPN1, okay, in your gene synthesized fragment, you need to use KPN1 as well, because after cutting, KPN1 sequence can like complement, okay. Or in the second possibility is, if you do not find any RE site in your expression vector, but you already decided a region that you want to use and continue with your gene interest, you can let us know. We do offer a service that do not use RE cloning. Okay, we can generate a vector, plasmid with vector sequence up to a point, continue with the with your gene sequence, okay, without RE site in between them. Okay, because our strategy most of the time is vector sequence, vector, then RE site in the vector, okay, then continue with the expression vector sequence. Okay, so I'm not sure whether I am answering your question, but let's say you have further question, you can voice it out. Okay, what are the charges on the expression vector? Okay, so in general, a gene synthesis service for a 1,000 bases gene sequence costs around RM 2,000. Okay, and the subcloning service normally costs maybe I think 400 to 500 RM like that. Okay, the good thing is you can actually get the expression vector as well. You do not need to purchase the expression vector separately. Okay, can GeneScript provide cloning into a gateway cloning vector? Because the thing here over here is okay, for this question, I actually never has any customer that use gateway cloning strategy before. I might actually need to check with GeneScript whether they are offering the gateway cloning. But gateway cloning supposedly is a easier cloning method. So yeah, I need to check with them to confirm that they are offering gateway cloning.
All right. Another question from Nohaida. What kind of software do you use to perform codon optimization? Can you synthesize gene that has high complexity? How about gene that has high GC content?
Okay, so for some of the question, I might not able to give you answer until you actually share the sequence to us. Okay, because the reason why we need the gene sequence is because GeneScript will actually analyze the sequence, because they actually sort of like guarantee the deliver sequence. Therefore, every time they will actually check the sequence, they will reply to us whether they are able to synthesize that sequence or not. Therefore, when you're asking whether you have high GC content, some challenges, I'm unable to tell until I send the sequence to GeneScript. They actually scrutinize it. Then after that, they will let us know whether they are able to do it or not. Okay, but and if you have high GC content, and if you are coding sequence, okay, I mean this high GC content part belongs to the coding amino acid sequence. We can always use codon optimization to optimize it to become less, to reduce the GC content. Okay. For the codon optimization, okay, we are using a software called OptiGene, which is actually created by GenScript. Okay, so OptiGene algorithm from GenScript. Okay, so what is the next question? Yeah, so therefore for the high complexity and also GC content, we will need to actually check on the sequences, and then we shall let you know whether we are able to offer.
All right. Another question from HL Wong. Do we need to include the flanking RE sites to be used for insertion in the sequence that we are sending to GeneScript?
Just let us know the IE site you want to use, or you can actually straight away include it. Okay, if you straight away include it, let's say just now in this particular example, okay, where is it? Okay, So you can just straight away include it. Okay, sorry, let me have my pointer. Okay, so you can just straight away include it in front of your sequence. We shall synthesize start from this part up to the end of the other part. If you would like to, you can do like this format. You can say I start with IE side, then continue with my sequence. Okay, so we will synthesize it from the IE side up to another IE side. Okay, so if you want to have any modification or IE side, you need to include in the sequence that you send to us, or you need to inform us. Okay, otherwise we will just synthesize the sequence based on the sequence that you submitting to us.
Yeah, thank you, Chi Hong. Ross Hariza is requesting for the paper from the Burgess Brown et al. Can Chi Hong share with Ross Hariza?
Because actually I haven't go to the paper yet, so I will try to look for the paper. Then if I get it, then I will share it to you.
Yeah, at the same time, can Ross Hariza leave your details over here, or you can contact our product specialist to leave your contact about this. Then we will try to share the link with you. Thank you. And the last question from Nohaida. Can codon optimization of the gene improve the protein activity in our target host cells?
Okay, protein activity, I don't think so. Why I'm saying this is because codon optimization did not change any amino acid sequence. Codon optimization only changed the nucleotide sequence. Okay, so therefore the acid sequence will remain the same before and after codon optimization. Okay, just that it helped for, let's say, the in-between process, the translation process. Okay.
May I know if there is any more question for our speakers, Chi Hong, today?
Yes, can I just ask through the microphone?
Can, sure.
Right. I'm not sure. I just came in late just now. Just wonder how big is the gene? I mean, do you have any limitation for the gene size?
Okay. We do not set a limitation, but we have experience of synthesizing the whole plasmid, because in some cases, the researcher, they actually, how to say, they found the full sequence of a plasmid, but they are unable to source the full plasmid. So the size of more than 10 kb. Okay, so we have the experience to construct the whole plasmid that with a size of more than 10 kb. But of course, okay, I cannot tell you that we sure can do it, because we will need to look at the sequence first, and then you can share it to us. Then we shall check, and then after that we shall able to reply to you whether we are able to pick up the task.
Yeah, I'm asking because some of the non-expression and expression vector, they do have like limitation to accommodate the size of.
Okay, understand. Okay, so because the limitation is not sometimes not because of our gene synthesis technique, but more to like after we deliver the plasmid to the user. Okay,
Yeah.
can you actually deliver the plasmid into your whole cell of interest? That will be a challenge, because if your plasmid is too big, sometimes we might having difficulty to try to transform or transfer it into the whole cell. So therefore, for GeneScript part, normally there are no problem to create it, but the problem might be, let's say when the user receive it, can they actually deliver it into their whole cell of interest?
Okay, one last question. So when you say that we need to send you the sequence, do we need to send the whole sequence or just a transcript of the gene?
A lot of times is actually okay. So this one is okay. A lot of times we only need the coding sequence. Okay. So let's say for human genes, a lot of times because they are like intron and all that, or they have like 5' UTR, 3' UTR, and all that. Okay. For heterologous expression, most of the time we only have the coding sequence. So basically, a lot of time we only start with ATG. Then after that, continue with the next amino acid, next, next. Okay. And then if you want to add any tags, if you want to have his tag, so we just add the his, okay, 6x. Or if you need some other modification or tags, we will just continue. And then after that, when ending, we will actually end with a stop codon.
Oh, so that means the mRNA of the sequence, this mRNA sequence.
The code, the CDS of the mRNA sequence. Because sometimes in the accession number, they give you like the full length mRNA, but the one that coding for amino acid is only like, let's say, starting from a certain length. Okay.
Okay. Yeah. All right. Thank you.
Okay. Thank you.
All right. Is there any more questions from the floor? Chi Hong, is there anything else that you want to cover up before we end our webinar session today?
Yes, I would like to thank everyone for your question and your participation. Sorry for your participation. Yeah, and then if you need any help, our friendly sales person and team will be there for helping you. If you have any question, you can always contact us, and we will try to answer and help out for the whatever that you require for gene synthesis or subcloning for expression. Okay, but unfortunately, sometimes, let's say when you're talking about like the interaction of the protein inside a particular cell, so that will be the real life situation. Sometimes it's not under human control. So those questions might need to try and error. Okay. We can only try to design the best. Okay. So therefore, the designing part will be quite time consuming and crucial, because when you already receive the plasmid, the price already paid. Okay. So therefore, you want to revert, then you realize there are mistakes. Okay, then it's already delivered, then it will be difficult. So therefore, we try to, let's say, look at the expression vector, choose the correct expression vector, and then choose the IE site correctly, and then try to design it nicely. So we should spend more time during that stage, actually. Okay, so you can always contact us for some discussion about how you want to design it, okay, to try to ideally get the correct sequence when we deliver it to you. Okay, but in any case, if let's say you found any modification that you require to modify after you receive the vector, no worry, because we will store the plasmid, okay, and then we will charge like modification fee, like the mutagenesis service that costs maybe 700 to 800 instead of charging you the full gene synthesis service that maybe cost 2,000 or 3,000. Okay, so it can be done in that sense as well.
Right, thank you, Chi Hong. So I think that's the end of our webinar today. And thank you so much for everyone for joining our webinar session today. If you have any inquiries, feel free to contact us at sales@nextgene.my, as I shared in the chat box, for any if you have any further questions. And also, we will send the recording of this webinar and the slides to the participants after the webinar session, so that you can watch it again at a later time or share it with someone who might be interested in this. Lastly, we hope that you find this webinar useful, and also we are seek for your cooperation to fill up the survey form as shared in the chat box. Your feedback will be valuable in helping us to improve our future events. Last but not least, stay safe, take care, and have a pleasant day ahead. Thank you very much for joining the webinar.
Thank you, everyone.